Hello and good morning everyone. Today we shall discuss about sample collection, anticoagulants and disposal of laboratory waste or hospital waste one by one. So let us proceed. So the first section is the sample collection, and among samples that are commonly used are the body fluids. So let us see some of the body fluids. These include cerebrospinal fluid, saliva, bile, gastric juice, milk, urine, blood. They also include semen, ascitic fluid, synovial fluid, pleural fluid, amniotic fluid, sweat, pericardial fluid. Now I've tabulated the various samples, how they are collected and what is the purpose of their collection. So we will discuss one by one. So we'll begin with cerebrospinal fluid, which is abbreviated as CSF. Now the requirement for collecting CSF is a spinal needle and sterile vials. The spinal needle dimensions are mentioned here. The most commonly used gauge is the 22 gauge needle. CSF collection is done by puncturing the subarachnoid space in the interspace between third and fourth or the fourth and fifth lumbar vertebra using a spinal needle under aseptic conditions. Now, after collecting the sample, it is transferred to a sterile vial and it can be sent for biochemical, pathological and microbiological examinations under various conditions. Now the second sample in discussion is saliva. For salivary sample collection, we need deionized water or demineralized water, clean and dry sterile test tubes or beaker. The procedure is first rinse your mouth and clean it of all the food particles and their remnants. Then take 10 ml of deionized or demineralized water and keep it inside the mouth for 2 minutes. Now while the water is inside the mouth, it needs to be moved gently with the help of tongue. All the secretions collected in this duration is now emptied or transferred into a clean test tube or a beaker with the help of a funnel if needed. This sample is analyzed for biochemical microbiological examinations. Salivary sample can also be checked for amylase activity on starch. The next body fluid in discussion is urine. For collection of urine sample, we need urine cup or a urine jar or a sterile cup. For 24-hour collection, we need a glass container having the capacity of 2 to 3 liters. The purpose in general for urine sample collection is to carry out biochemical, microscopic and bacteriological examinations. Now there are various types of urine sample collections. Let us see each one. The first one is random specimen. No specific time for collection is needed. It is the most common type of urine sample collected. It can be collected at any time of the day and it is generally used for routine screening tests. The second sample is the morning sample, which is the first urine sample in the morning, which is most concentrated and therefore it is ideal to carry out the pregnancy test and the microscopic examination of urine. The clean catch midstream sample is another urine sample in which the first few ml of urine are discarded and the remaining volume is collected, and generally this sample is used for culturing the urine. The next sample is 24-hour collection, and the entire urine sample that is passed throughout the day and night and the next day first sample is all collected in a single container The container must have a preservative so that no bacterial growth takes place during the collection time. Such sample of 24 hours is generally used for quantitative and qualitative analysis of various substances present in the urine. This is generally done to assess the renal functions. The next sample is the postprandial sample, which is collected 2 hours after the meal, and it is done to determine the glucose levels, generally to check diabetic monitoring or diabetic control in patients. Another kind of sample is a suprapubic aspired sample, which is done with the help of needle aspiration, and this is done to obtain sterile urine. It is generally seen in pediatric cases. Now the fourth body fluid that we are going to discuss is the gastric juice. So in order to collect gastric juice, we need Ryle's tube, syringe, sterile and clean beakers. Now what is Ryle's tube? Ryle's tube is a flexible plastic tube with a diameter of 4 mm and it is 22 inches long. The aspiration of the gastric juice is done by a syringe through the Ryle's tube from this end. So we also need a syringe for the collection of gastric juice along with the Ryle's tube. Now the purpose of collecting gastric juice is to carry out biochemical and microbiological examinations such as for the diagnosis of stomach and duodenal diseases such as peptic ulcer, gastric carcinoma, gastritis, pernicious anemia, hyperacidity status in Zollinger-Ellison syndrome. It may also be used for differential diagnosis of gastric and duodenal ulcers. With the help of gastric juice, one can also find out the basic acid output and the maximal acid output during the day. Now, the next important biological fluid is blood, and the blood sample collection needs a following items such as tourniquet, cotton, 70% isopropyl alcohol, syringe, needle. One can also use lancet for capillary collection or capillary tubes. We need needle cutters, waste disposal bags and vacutainer tubes. Now generally we need blood samples for various biochemical, pathological and microbiological examinations. So there are different types of blood sample collections depending upon the site of collection. So let us discuss the various types. So we begin from the arterial puncture. So whenever an arterial blood sample is collected, it is done through arterial puncture. The site for the site or the favorable or preferable arteries may be the radial artery, brachial artery or the femoral artery. Generally, arterial collection is done to carry out blood gas analysis and electrolyte analysis. Now, if the blood sample is collected from the veins, it is done with the help of venipuncture, and the preferred vein is the median cubital vein. Generally, venipuncture is done to carry out various estimations such as enzyme, electrolyte, various metabolites and hormones present in the blood. One can also carry out the find out the hemoglobin concentration and various cells present in the blood. The third kind of blood collection is the capillary collection. The preferred site is the third and the fourth fingertip. The other sites will be mentioned in the next slide. Generally, capillary collection is done for sugar, urea and blood gas analysis, or whenever the sample requirement is very less, capillary collection can be done. For example, when we want to determine the blood group of a person. Capillary collection is done. Now, these are the various sites of blood collection. So let us begin with the capillary collection. So you can see that these are the places, that is the third and the fourth finger, that is the ring and the great finger, from which fingertips, from where the capillary collection can be done from hands, whereas from the heels, infant's heel also, it can be collected from these two sides, and the earlobe also may be used for the capillary collection. For capillary collection, we need a lancet. The lancet has a needle which is used for a sterile needle, which is used for piercing the capillary or puncturing the capillary. The next is the venipuncture. For a venipuncture, the most preferred site is the median cubital vein. So generally when we are selecting the site, we have to take into consideration the surrounding of that particular blood vessel, and also the diameter and the thickness of the blood vessel. So based on all these criteria, the median cubital vein is the most preferred site for blood collection, or the venipuncture. Now the next is the arterial puncture, which is done from the brachial artery or the radial artery, and it can also be done from the femoral artery. So these are the different sites of blood sample collection. So this is a needle. These are the parts of a needle, which has a hub, the cannula or the shaft, and the bevel. So this pointed region is known as the bevel, which is inserted for sample collection of blood. Now every needle has a gauge, and the gauge is actually the diameter of the needle. The routinely used gauge is the 21G. In children, the 23 gauge is used. The larger the number of gauge, the smaller is the needle. As you can see here, this is the smallest number and this is the largest, and the size of the needle is inverse to the gauge. The needles are color coded, that is the hub of the needle is color coded. It ranges from the size of 16 to 25, and the most commonly used size is the 21G. Now this is a structure of the syringe. This is a routinely used syringe which has a plunger. It has a barrel in which the blood sample is filled. This plunger helps to limit the amount of blood inside the syringe, and this is the needle. The needle, as you can see, has a bevel here, and this bevel actually has a lumen, and through this lumen the blood flows into the barrel of the syringe. Now this is a vacutainer system which operates under the vacuum. This whole system consists of a two-sided needle, and from this system we can collect multiple samples. It has a tube holder, it has a vacutainer tube, and when all these three are assembled together, this is how it looks. And every time from this very needle we can collect the sample in different tubes. So every time all we need to do is remove the replace the tube and place another tube over here. So this is how the vacutainer system. So in order to carry out blood collection, first we need to select a site, and after selecting the site we need to disinfect it with the help of spirit or alcohol, and generally we use 70% ethanol. Then we use a tourniquet which helps us to which helps the vein to make it prominent, and after tying the tourniquet, disinfecting the area. This is how the sample is collected. Now after collecting the sample, it has to be transferred to various vacutainer tubes. As you can see, these are the most commonly used vacutainer tubes, and each vacutainer tube has a different colored cap, and the different colored cap signifies something which we will discuss in the next session. So currently you just have to remember these different 5 colored caps, that is the light blue, red, green, lavender and grey. So now we come to the next section, that is the anticoagulants. So these are the different anticoagulants or additives that are commonly present in the vacutainer tubes. So let us see some details how these additives work and at what concentration. So we begin with heparin. 2 milligram per ml of blood is needed, and it acts by preventing thrombin formation. The next additive is sodium citrate, which is also an anticoagulant, and it is needed in the concentration of 3 to 6 milligrams per ml. It forms calcium complex and prevents blood clotting. The third anticoagulant is EDTA, that is ethylene diamine tetraacetic acid. It is required in the concentration of 10 to 20 milligrams per ml, and it is a calcium chelator, so it chelates calcium ions. The next additive is in combination. It has sodium fluoride and potassium oxalate. Both of these additives are present in the same tube, and their mechanisms are different. Sodium fluoride is used in 10 milligrams per ml of concentration, and it is generally used as an inhibitor of glycolysis, so that true estimation of blood glucose is done. Fluoride acts by competing with magnesium ions, which is a cofactor for enzyme hexokinase as well as enolase. So fluoride inhibits both these enzymes, and that is how it prevents glycolysis done by the blood cells. Potassium oxalate is used in 2 to 3 milligrams per ml concentration, and it is a calcium binder, and therefore it prevents blood clotting. So both these additives are added in a vacuity when glucose has to be estimated. The last one is a clot activator. It helps or enhances the blood clotting in the vacutainer tube, and this is how these different additives work in different tubes. Now the choice of these tubes will vary. For example, if we want to estimate calcium, since EDTA chelates calcium, we should never collect blood sample for calcium estimation in an EDTA containing tube, because it will give a false low value of calcium. So depending upon what we want to analyze, we should choose the additives present in the vacutainer tubes. Now the next section will tell you about the order of draw, and this order of draw is generally to avoid cross contamination whenever multiple collections are needed from the same site. So the blood is drawn and collected in tubes in a specific order. Now this order is based on the different types of additives present in the tube or different types of anticoagulants. This order followed is universal, and it is known as the order of draw. So let us see what is the order. So the first, whenever we are multiple collections are needed from the same site, this is the order that is followed. That is, first is citrate, then we go to the Then we add the sample in the vacuity containing clot activator, followed by heparin, followed by EDTA, followed by fluoride. So the last vacutainer always will be fluoride whenever multiple tubes are needed. Now after having collected in the tube, the blood sample has to be centrifuged for 10 minutes at 3000 rpm. So when it is centrifuged for 10 minutes, before 10 minutes, this is how it looks, and after centrifugation, these are the three layers that are formed, as you can see. So we'll begin from the lowermost layer, which is dark red in color, and that is because of the red blood cells, followed by a light red or pinkish color or whitish layer. This is known as the buffy coat, and this buffy coat or the white layer is very thin, and it consists of white blood cells and platelets, and the topmost layer is a clear transparent fluid, and this fluid is known as plasma or serum, depending upon the additive present in the tube. Now the last part of the topic is about disposal of laboratory. So there are different types of laboratory wastes, and these need to be disposed because any hospital waste is infectious, and so before disposing we need to carry out a few steps. So let us see what are the steps. The first step is of segregation. Segregation is done in color coded bags, which we will see in the forthcoming slide. After segregating the hospital waste into different categories, they are disinfected. Disinfection is done with 1% hypochlorite in liquid or by autoclaving for 20 minutes between 12 to 20 lb per inch square pressure, or with 10% hypochlorite for heavily contaminated items. So after segregated in color coded bags, these items will be disinfected, and then finally they will be taken to the disposal area. The liquid waste generated will be disposed of in the laboratory drain after disinfection, and the solid waste will be disposed as per hospital waste disposal system and the guidelines for it. So how is the hospital waste disposal done in different color coded bags? Let us see. Now these are the four color coded bags. As you can see, this is the dustbin: yellow color, red, white and blue. Now each dustbin has a symbol. This symbol is a biohazzard. Inside each dustbin lies a plastic bag which is also of the same color to that of the bucket. So both the bucket and the bag are of the same color, and both of them have the biohazzard symbol. Because we are aiming to dispose of hospital waste, and all hospital waste is considered infectious or hazardous, so they have the symbol of biohazzard. Now let us see what are the different wastes that can be disposed in different color coded bins. So in the yellow color bucket, we can dispose of various anatomical parts such as placenta, pathological waste, cotton waste, dressing material, beddings, body fluids, cytotoxic expired and discarded medicines, microbiology, biotechnological laboratory waste, face mask, cap. In the red colored bag, we can dispose of syringe without needle, IV sets, catheters, gloves, urine bag, dialysis kit, IV bottles. So it generally includes the plastic wastes, hospital waste. Whereas in the white bags, we dispose of the metal sharps, such as needles, syringes with fixed needles, blade, scalpels, lancets. These are all disposed in the white bag. While in the blue bag, broken glass, ampules, and lab sites are disposed, and some metallic implants such as nails, metal body implants, and scissors are also disposed in the blue bag. So this is how the color various hospital wastes are disposed in color coded bags, and these colors are universally decided. So anywhere in the world you go, the same things will be disposed. Hospital waste will be disposed in the these color coded bags. So a biological hazard is defined as a hazard that refers to biological substances that pose threat to the health of living organisms, primarily to that of humans. This can include samples of microorganisms, virus, or toxin from biological source that can affect the human health. Now, there are various risk group classifications based on the community risk it has. So let us see the risk groups. There are risk group 1, 2, 3 and 4. So there are 4 risk groups classification. The first group has no or low risk. It is unlikely to cause animal or human disease. It generally consists of non-pathogenic agents. The community level risk is nil or low. Examples of such organisms are non-pathogenic strains of E. coli. The second risk group includes the individual risk is moderate. It includes pathogenic organisms for human beings. Treatment and preventive measures are available if somebody gets infected, and there's limited risk of spread of infection, and the community risk is low. These include Staphylococcus aureus, hepatitis A, B, and C viruses, HIV, Plasmodium falciparum. The third group has high individual risk. They consist of pathogenic organisms. They cause serious diseases. If anybody is exposed to these uh, organisms, the individual has serious disease. Effective treatment and preventive measures are usually available. Community risk is low, and it includes rickettsia, Brucella, chikungunya, yellow fever virus, etc. The last and the highest risk group containing organisms is kept in the fourth group. It has the highest individual risk. It is lethal. It's pathogenic agent, readily transmittable through direct or indirect means. Effective treatment and preventive measures are usually not available, and therefore the community risk is very high, and it includes Nipah virus, Hendra virus, Ebola, and the Marburg viruses. So these are the different risk group classification depending upon the organism and the individual risk and community risk, that is that they uh, that they are capable of causing. Now, if any lab is working on all these different types of organisms, the lab will be divided into different sections based on the risk group, and they will follow suitable guidelines whenever they are working on such organism. Especially the high risk group organisms will have to follow very precautious steps to carry out the analysis or the various tests pertaining to these viruses. Now, these are some commonly used laboratory symbols like animal hazard, sharp instrument hazard, heat hazard, glassware hazard, chemical hazard, electrical hazard, eye and face hazard, fire hazard, biohazard, laser radiation hazard, radioactive hazard, and explosive hazard. So these are various commonly used laboratory symbols that are present in different containers you generally see in a laboratory or in hospital area. Whenever you see any one of these symbols, it just tells you about the content present inside that box or inside that bag which has the symbol. For example, a box has this symbol, so you must understand that that box contains certain chemicals which are hazardous. Thank you.